Hi, my name is Juliana Bablak, and today I will be going over the evolution of DNA and crime solving. So what is DNA? DNA, or deoxyribonucleic acid, is a molecule that carries out our hereditary information. The double helix allows for replication, and below I have shown a picture of the replication when the double helix is unwound. So how DNA works. DNA carries the instructions that are needed for an organism to grow, live, and reproduce. To do this, DNA sequences are converted into messages that are used to produce proteins. DNA sequences are in the order of 4 base pairs that make up the DNA molecule. These are known as adenine, cytosine, guanine, and thymine. Each sequence that holds instructions to make a protein is called a gene. The journey to gene to protein is intricate and strictly monitored within each cell. There are 2 major steps, which I have shown below in a picture, called transcription and translation. So DNA goes through transcription to become RNA, which is then translated to form amino acid chains, and then the chains are then folded to create a protein. So the history of DNA and crime solving. This is called forensic DNA profiling, and DNA analysis was first used in 1987. This was used to find Colin Pitchfork, who tried to avoid DNA testing as they were collecting fingerprints. He was eventually found and admitted to killing 2 girls. So the next thing is gel electrophoresis, which is isolated and separated DNA fragments that are sorted by size and charge. The DNA sample is put into a well at one end of a gel, and then an electric current is applied to pull the DNA through the gel. The small fragments move through the gel faster than the large ones, and the gel is then stained with a DNA binding dye, and the DNA fragments show up as bands. The next type is STR, which is short tandem repeats, and this was introduced in the 1990s. This is when DNA has regions where short sequences of bases, which were covered in the previous slide, are repeated multiple times. The repeats are found in many spots throughout the genome. The exact number of repeats varies per person, and in forensic science, we use these STRs to identify individuals. Next thing is PCR, which is polymerase chain reaction. This technique greatly advanced DNA analysis techniques. In PCR, enzymes amplify the amount of DNA in a sample by copying it many times, which makes it easier to detect. Thanks to PCR, DNA profiles can be collected from much smaller samples. The next thing is a profile archive. One that you may be familiar with is CODIS, which is the Combined DNA Index System. This was established in 1998 by the FBI. CODIS includes state databases as well as the national database that's known as the National DNA Index System. Only accredited government laboratories can submit profiles to the national database. The next type that we have is probabilistic genotyping. This uses statistical software for analyzing complex DNA evidence. This allows analysts to recover DNA evidence from samples that they had previously declared inconclusive. The next type that we have is next-generation sequencing. This is something that scientists are in the early stages of evaluating. So the first type is DNA sequence info, and they are currently working on ways to use the DNA sequencing information to establish features of a perpetrator's physical appearance, such as eye, hair, and skin color. There's also epigenetic markers on DNA, which there are studies going on to see if epigenetic markers on DNA can be used to determine a suspect's age and the type of bodily fluid a sample came from. So how DNA is found at crime scenes. Fingerprints were once the benchmark for connecting suspects to a crime scene. Today the new benchmark is DNA because it can be found virtually anywhere. Even if a criminal happens to wear gloves, DNA still may be left behind at the crime scene. DNA is found in hair, saliva, blood, semen, skin, sweat, mucus, or earwax. It only takes a couple of cells now to have enough DNA to ID a suspect. and great care must be taken when collecting evidence so as not to contaminate it, because big mistakes can happen when the evidence is analyzed and it's been contaminated. So there are many common sources of DNA. They can come from a weapon, a hat or a mask, a facial tissue and cotton swab, toothpick, cigarette butt, bottle, coffee cup, and even a postage stamp. It can come from a used condom, as well as bed linens and towels, fingernails, dirty laundry, and eyeglasses. So using DNA today. DNA is used to solve crimes, exonerate the wrongly convicted, and solve cold cases. DNA profiles today can now be gathered from single cells, whereas earlier on, it was necessary to have large samples of blood or bodily fluids to even get a profile. The techniques of DNA collecting have greatly evolved, and DNA databases have helped tremendously with this. There is now a better understanding of how DNA is transferred and the risks of contamination that come with it. There are 4 different types of DNA that can be used to solve crimes. There's mtDNA, y-DNA, autosomal DNA, and nuclear DNA. Nuclear DNA is examined in evidence that has bodily fluids, skin cells, bones, and hairs that have the tissue at the root end. Nuclear DNA has the ability to identify or exclude an individual as a contributor of the DNA evidence that was obtained and analyzed at a crime scene. This is inherited from both parents, and it's most ideal when trying to ID someone in a crime. The next type of DNA is mtDNA, or mitochondrial DNA. This is maternally inherited and is used for obtaining information when nuclear DNA analysis is not attainable. mtDNA is used when obtaining information from old evidence. This is usually associated with cold cases or small pieces of evidence that have little biological material. It can be compared with other remains or samples from other maternally related individuals as well. Now we have y-DNA. Y-DNA is the Y chromosome DNA. It is present only in males and is paternally inherited. This can help track paternal ancestors, and this is used when autosomal DNA is not informative. This is mostly used in sexual assault cases when the suspect is a male. Now we have autosomal DNA. Autosomal DNA is DNA from any of the 22 pairs of autosomes in the genome. This is not including the 23rd, as this is a pair of sex chromosomes. Autosomal DNA is most used when solving crimes because, excluding identical twins, no 2 people have the same autosomal DNA. And with all of this comes wrongful convictions. The majority of wrongful convictions are associated with serology, which is the identification and characterization of biological samples. This can also be associated with microscopic hair analysis, which is to see if hairs match. This is usually found in trace evidence, which is transferred during the act of a violent crime. We also have bite marks and ABO blood typing. With ABO blood typing, it's usually in how the information is interpreted or conveyed, and misinterpretations can be made by non-scientists. Forensic science can be a part of wrongful convictions, but there are usually several other contributing factors as well that are not linked to science that can cause these wrongful convictions. A situation you may be familiar with is college student Amanda Knox, who was convicted and then eventually acquitted in the murder of her British roommate Meredith Kercher in Italy. This had to deal with contaminated evidence that was found at the scene because they did not take enough care when collecting the evidence. For my peer-reviewed article, it talked about the future of forensic DNA analysis. They talked about faster results, which they said there are limits, but as PCR amplification gets faster, the DNA separation and detection part may be the rate-limiting step. So PCR amplification is a combination of polymerase biochemistry, primer binding kinetics, and thermal cycling device. Higher sensitivity and information content is another thing. There might be more information from expanded sets of STR loci and supplemental genetic markers. There's also might be more in-depth information from sequence analysis of alleles. The next part is stronger conclusions from samples. Challenging samples are things like degraded DNA samples or uninformative PCR profiles, as well as complex DNA mixtures. and they're hoping with mini STRs or SNPs, this can help recover information from a damaged DNA. Mini STRs are mini short tandem repeats, and SNPs are single nucleotide polymorphisms. The next part is familial research to help identify relatives when the PCR profile is incomplete. And the last part is hopefully getting to the part of predictions of visible characteristics. Now, this is how it all connects to cellular and molecular biology. We've talked about DNA and how it is structured and how it works. We will also talk about DNA sequences, genetic codes, and genes, which all work into how DNA can be used to solve crimes. We also know that DNA is a type of nucleic acid. Now, because DNA is so complex, we still have a lot to learn about it and how it can be used to continue to solve crimes, as well as in many other areas in the sciences as we uncover more about DNA. This is including the value of not only coding proteins, which are genes, which only makes up a small part of the genome, but also the non-coding proteins, which are becoming more important than we may have once thought. So these are my works cited, and the last thing I have is a TikTok that my friend sent me about how DNA is structured and what it is made up of. And with that, I hope you enjoy my presentation, and thank you for listening.